Hello, and welcome to today's lesson on protein synthesis. Now, what is protein synthesis? Protein synthesis refers to the construction of proteins by the living cells and is comprised of two parts, transcription and translation. But before we jump into the process, first let's start with a quick review on DNA. Both DNA and RNA consist of three parts: a 5-carbon sugar, phosphate, and nitrogen bases. The sugars and the phosphates alternate to create the backbone, as I've represented with construction paper, right here. And the variable bases carry hereditary information, as I've represented with these pom-poms. Your DNA is composed of 4 different variable bases called adenine, guanine, thymine, and cytosine. And if you see here, we have just a little key to represent each one on our DNA here. As you see, if we put both of them together, your A will only bind to T, creating 2 hydrogen bonds, and C will only bond to G, creating 3 hydrogen bonds. Now, the sequence of these 4 bases can be coded to translate to make any protein in your body. Now, before we move on, we also have to talk about RNA. Now, there are 3 types of RNA, and the first one that we're going to talk about is the mRNA messenger. So here we have another example, but this time we have the base being a different color to represent that this is the mRNA. So this essentially carries the photocopy of the DNA and the blueprints of the nucleus of the ribosome to make a protein. And this will be later read in groups of 3 called codons. If you may notice, compared to our one DNA strand, you may see that there are pink pom-poms instead of the yellow. This is because mRNA instead has uracil instead of thymine. But it will still mean that uracil will bond to A. I'll put that back here. The next part of RNA that we're going to talk about is the tRNA transfer. We'll see this a little better in detail later on, but here I have my tRNA carriers. And these are what is going to hold on to our amino acids, and when the codon lines up with the anticodon, the tRNA drops off the appropriate amino acid off on the chain. So later we'll see I have our amino acids here represented as little dinosaur stickers, so these will later carry on and drop these off. Then the next type of RNA we'll be talking about is the rRNA, which is the ribosomal. Again, we'll get into this in better detail later. Here I just have a ribosome to show here. All right. So what about protein? Well, protein is made from individual amino acids that join together in long chains. There are 20 different amino acids that create all your proteins. And here I've represented this through, I have a bunch of these different amino acids as little dinosaurs. All proteins always start with the start codon, AUG, and will also always have a stop codon ending in UAA, UAG, and UGA. So now that we have a little bit of base knowledge, let's start with the first step of protein synthesis, which is transcription. All right, so we're looking at transcription. Now, the method that I use to remember this process is by looking at the name. Transcription sounds a lot like transcribe, to write down or copy something that someone is saying. And that is exactly what we're going to be doing here. So as you see, we have our DNA that's in your nucleus. So what begins to happen once these are attached, they begin to separate. In this process, mRNA will begin to copy itself in between, but instead of T, it will use uracil. So if you take a look at the secondary strand here and the mRNA, you notice that they are completely alike. They match. The only difference here is going to be between the thymine and the uracil. So that is essentially what is happening in transcription. Your mRNA comes in and copies the leading strand of your DNA. So now that we have our photocopy, we can move on to the next step of protein synthesis, which is now translation. So once we have our photocopy, mRNA begins to snake out of the nucleus, comes out here. Your DNA will slap shut again to protect it. And it will come out of this nucleus and it will be floating into your cytoplasm until it reaches a ribosome here. And again, the method I use to remember this process is by looking at the name. Translation happens between two different languages, and that is essentially what happens in this process, where DNA language transforms into the protein language. So once we reach the ribosome, it will bind to the top and begin pulling the mRNA through, reading it in groups of 3 called a codon. In order to translate the 4-letter DNA language into the protein language of 20 amino acids, recall that we first have to find our start codon AUG. So from here on, the sequence of these letters will correspond to the tRNA's anticodon carrying a specific amino acid. Now here I went ahead, I just created a bit of a sheet so we don't have to look at this. And as always, if you recall, the start is going to be our AUG. So here I'm going to place our start codon right there. Our next one is going to be a GAU. So our tRNA is going to come over, pick up the amino acid, and drop it off at the appropriate spot. Let's look at our next one. UUG. So again, here comes our tRNA, plugging it in, and then leaving later to be recycled. Next one we have GCA. We have a yellow dinosaur here. We have next UGG. Here comes purple dinosaur. So again, we read these in groups of 3 called codons, and our tRNA will then carry the correct amino acid over, pop it in like a socket, and then leave. This will continue later until we reach one of the 3 stop codons. And here I've just wrote UGA. So let's enter our stop codon there. Once we reach this end, the amino acid chain will begin to fold into its protein shape and become ready for transport. So yay, we have just made our complete protein. I'll move this all down. So now that we know a bit about protein synthesis, let us talk now about mutations. Mutation refers to the changing of the structure of a gene, and you've probably commonly heard about this in things like Teenage Mutant Ninja Turtles or a popular movie series, X-Men with mutants. Now, mutations can occur for 2 reasons. The first is simply from making mistakes in your DNA. The first is simply from making mistakes in your DNA because your body makes mistakes regularly. And the second occurs from environmental factors with known carcinogens. This can be anything from UV rays, chemicals from smoking, or even radiation. However, in this lesson, we're only going to be learning about single base mutations, and which include 2 main divisions. So first mutation that we're going to be talking about is called point mutation. All right, so in point mutation happens when a single base in the DNA gets substituted. So we're going to start here. I'm going to go down the 7th one. 1, 2, 3, 4, 5, 6, 7. I'm going to change thymine here to a D9, which then means that our RNA will have to change as well. And usually A binds to T, but remember, mRNA does not have any thymine, instead it has uracil. So let's change this one to a pink. So now that we have a substitution, we can again take our tRNAs and plug them in. So we're going to start with our AUG, of course. It's our first one. GUA we have for our second one. We have CGG, and we have the CGG there. We have UUU, and now instead of what was AGG will now be UGG. So in this case, when we substitute a certain base, sometimes your genes can be altered and sometimes they will not. In the next case, we have frame shift mutations. So instead of substituting a hereditary base, we are going to be deleting one. Here, let's again delete the 7th one. 1, 2, 4, 5, 6, 7. And let's set up our appropriate amino acids here again. So again, we're going to start with our AUG. Next we have GUA, CGG, UUU. And now that we have deleted this one, we're going to go on to GGU. So let's find our next one here. So instead, what we had this green monster or green dinosaur in an egg, it will now switch out with this green and pink dinosaur here. So when we have mutations, they can occur in two different ways. First, we have what we call silent mutations. And this is what happens when there are changes to the bases, but the amino acid stays the same. As you can see on some of these amino acids, there are multiple codes for certain things. So even, say this we have is CGG, even if we were to replace this, say with another C, CGC still codes for the same amino acid. Therefore, there won't be much of a change. Secondly, we have missense mutations. Now this one actually changes the protein shape, of which we just saw in the last one. We went from, AGG here, and once we deleted it, we now have a new amino acid of GGU, which was replaced here. Perfect. Now that is the end of our lesson on protein synthesis.